Welcome to the 2021 American Academy of Neurology Annual Meeting Top Science Press Conference. The American Academy of Neurology Annual Meeting is the world's largest gathering of neurologists and neuroscience professionals, and this year attendees will gather virtually from all over the world, April 17th through the 22nd. My name is Andy M. Holt, and I will be your host during this press conference. At this year's meeting, more than 2,000 scientific abstracts are being presented by researchers sharing the latest scientific advances in brain disease. You'll hear about 3 of them today. We would like to thank members of the press for joining us for today's press conference. Today, Dr. Natalia Rost will be presenting her choices for the top science at this year's meeting. Dr. Rost is the chief of the stroke division at Massachusetts General Hospital, professor of neurology at Harvard Medical School, and the chair of the American Academy of Neurology's Science Committee. It is the AAN Science Committee that oversees the selection of the science being presented at the AAN Annual Meeting. Joining her are 3 authors of abstracts selected by Dr. Rost to be featured at today's press conference: Dr. Thanh Nguyen of Boston Medical Center, Dr. Sabrina Paganoni of Massachusetts General Hospital, and Dr. Michelle M. Milke of Mayo Clinic in Rochester, Minnesota. After each author presents their abstracts, Dr. Rost will ask a few questions. Then we will take questions from the journalists via the Q&A function. Please remember when posting your question in the Q&A to identify yourself and your news outlet. We will ask that you use the chat only if you're having technical issues. We will be calling on reporters with questions and ask that you unmute yourself if called upon. I would now like to introduce Dr. Natalia Rost, who will make some opening remarks about this year's meeting and her selections for top science. Dr. Rost.
Thank you, Andy, and hello, everybody. It is my great pleasure to welcome you all to our first virtual Top Science Conference. And for those of you who are new to our conference, we're pleased to meet you. For those of you who are returning, it's great to have you back again. And while I miss seeing you all in our usual press conference room, I'm grateful to have an opportunity to speak to you again after a year of hiatus caused by the COVID-19 pandemic. While this has been a trying year for the entire world, specifically, it's been immensely difficult for the medical community and the community of scientists, for many of whom their life work being rudely interrupted by the virus. But this also has been a year of tremendous triumph of science, with the extraordinary breakthroughs in diagnosis, vaccine development, and treatments for COVID-19 infection. And despite all odds of last year, with over 2,000 original abstract submissions to our virtual annual meeting this year, it's telling me that the resilience, remarkable creativity, and academic drive are alive and well among my neuroscience colleagues. And so, as we've done in the past, I will present to you today the 3 original studies which I selected as the exemplar of the AAN's Top Science. And for those of you who are returning to our event, you might remember that I like to select a theme for this conference. And so today, I think our theme will indeed be the celebration of science. And we have a lot to celebrate: a truly global collaboration of stroke neurologists in the heat of the pandemic to address the widely discussed issue of COVID and stroke, an innovative clinical trial design to bring about a cure for ALS, and a formative study that, in search of diagnostic breakthroughs, advances a promising biomarker of cognitive decline. All of these are examples of top science at the 2021 AAN Annual Meeting and a reason for us to celebrate science. But I also would like to highlight the fact that the 3 distinguished presenters today also represent the leadership by women in science, which is rapidly gaining momentum in neurological research, and which is another reason for us to celebrate and rejoice. So with that preamble, I'd like to start the official portion of our program today, Andy. Thank you.
Thank you, Dr. Rost. To begin our celebration of science, I would like to introduce Dr. Rost's first guest, Dr. Thanh Nguyen. Now, Dr. Nguyen will present her abstract titled Global Impact of COVID-19 Pandemic on Stroke Care and Intravenous Thrombolysis. This abstract is no longer embargoed. Welcome, Dr. Nguyen. Please share with us a summary of the findings of your research.
Thank you very much, Andy. Thank you, Dr. Rost, Ms. Tessman, the AAN neuroscience community for this great opportunity. Frankly, I wasn't expecting this, but we are very humbled to share our work. I would also like to highlight my gratitude to my co-PI of this project, Dr. Raul Noguera, our lead statistician, Mohammed Qureshi, and our many collaborators across the globe who contributed data to help us better understand the intersections of the COVID-19 pandemic and stroke care. As background, early in the pandemic, we learned about reports of declines in stroke care and acute stroke treatment across local, regional, and national studies. However, these reports originated from highly resourced countries such as the United States, Canada, China, France, Germany, Spain, and Italy, amongst many others. However, as the pandemic takes on a global spread, we sought to understand the effect of the COVID-19 pandemic on stroke care, acute stroke treatment with intravenous thrombolysis, and intravenous thrombolysis transfers across centers across the globe in low and middle income countries, primary stroke centers, and also centers with low COVID-19 burden. We defined the pandemic period as March 1st, 2020 to June 30th, 2020, and compared these stroke metrics with two control periods immediately prior and 1 year prior to account for seasonal variations. We learned that overall, across 6 continents, 70 countries, and 457 stroke centers, of which approximately a third were primary stroke centers, 2/3 comprehensive stroke centers, that there was a global decline in every stroke metric that we studied. Stroke hospitalizations dropped about 11.5%, and IV thrombolysis by 13.2%. We also did a recovery analysis. We compared the later versus the earlier pandemic months, the later months being May and June 2020 compared to the early pandemic months being March and April 2020. And we saw that recovery was seen across all strata of COVID-19 hospitalization burden. However, the gradient of recovery was less good in hospitals that had higher COVID-19 burden than hospitals that had lower COVID-19 burden. We also noticed that recovery was also higher in hospitals with baseline high volume stroke admissions and also in comprehensive stroke centers compared to primary stroke centers, suggesting that in the later months, patients with a higher degree of acuity potentially were seeking stroke care. Last, we also looked at the intersections of concomitant stroke and COVID-19 diagnoses. And across about 120,000 COVID-19 hospitalizations, we learned that the rate of stroke was estimated at 1.5%. And then across approximately 52,000 stroke hospitalizations during the pandemic that were surveyed, that the rate of SARS-CoV-2 positive status of these patients was estimated at 3.3%. So we thank you for your attention, and I'm open to questions at this time.
Thank you so much, Dr. Nguyen. This study is a true representation of a global collaboration, perhaps one of the most expensive across the multiple continents that I've ever seen. And having worked on some large collaborations, I wanted you to let our audience know how many authors you actually have on your paper.
As we committed to representing authors who contributed data, we had about more than 450 authors on the paper to represent approximately 457 stroke centers. And so figuring out a journal and a landing zone for this paper was not obvious, but we were very pleased that the Neurology Journal decided to accept our paper.
Yeah, I would like to commend you on the tremendous effort of writing a paper with almost 500 of your closest friends. And I wanted to ask you, what is striking to me that this paper is written in, right in the hit of the pandemic. And obviously to organize this has been not a simple task. But it must indicate that there is already a global network of stroke experts collaborating readily on that. Could you elaborate on how prepared this infrastructure was to kind of go into this project? And what do you think it signifies for future studies in stroke?
That's an excellent question, Dr. Rost. Obviously, we had the help of multiple societies contributing to this global effort. So the Society of Vascular Interventional Neurology led this charge along with my co-PI, Dr. Noguera. The Middle East, North Africa Stroke and Interventional Neurotherapeutics Organization contributed data from North Africa and the Middle East, and we had a great representation from this society. And we also had help from the Japanese Stroke, Vascular, and Interventional Neurology, led by Dr. Hiroshi Yamagami. And then in several countries, we found leaders who had stroke data ready to go. For example, the Czech Republic had lots of stroke data on stroke admissions and intravenous thrombolysis ready to go, with a little bit of verification on our end to make sure the data was complete. But we sought leaders across the globe, and we were very impressed by how organized stroke care was in several pockets of the world, especially Eastern Europe. I was pleasantly surprised, and also China, a great network there led by Dr. Zhongming Qiu.
Yeah, it tells me that the future of stroke treatments is bright in the way that there's a lot of a community of neurologists, stroke neurologists standing by ready to implement these studies as soon as we have treatments. Let me ask you another question. Obviously, stroke has been in the news from the earliest days of the pandemic, and there's been a lot of debate, back and forth debate. There's a lot of kind of a press attention, and then also internal data reviews that have been ongoing with regard to stroke incidence and COVID, etc. You are, however, showing the decline in stroke care. Do you think, and do you have data that demonstrates the severity of stroke affected by the pandemic in this particular work, or are you working on something in the future to be published? And do you also look at the stroke subtypes?
In this particular paper, we did not look at the severity grades of stroke because we had to think, if we're going to get a global study, what is reasonable data to ask from our co-authors. However, there have been many publications you've probably read as well, where if you have COVID-19 with a stroke diagnosis, that the severity of disability and mortality tends to be higher, and that's been published in several papers that have already been released.
Thank you so much, Dr. Nguyen. The paper now is open for questions.
Thank you very much, Dr. Rost and Dr. Nguyen. We will now take questions from journalists. Please post them in the Q&A area of Zoom, and remember to identify yourself and your media outlet. If we call on you to ask your question, please unmute yourself and ask your question. While your video will not be activated, the audio of your voice will be live. And I will turn this to Renee Tessman to bring forward the press in attendance.
Thank you very much, Andy. Yes, I would like to call on Lynn Peterson from Trends in Neurology. Lynn, if you can click on the box to allow yourself to unmute, then you can ask your question.
Actually, it's Trends in Medicine.
Sorry.
So not just neurology, although neurology is important. So today the news is about blood clots, including brain clots with the J&J vaccine. And yet there are multiple hundreds of cases, I think, of low platelet counts and ITP in the FDA's VAERS database with the Pfizer and Moderna vaccines. And so is there, how do we know that similar cases of thrombosis and ITP haven't already occurred in the senior population 2 or 3 months ago in the nursing home populations where fatalities may not have been fully adjudicated? So I know this is not the purpose of your study, but maybe you can shed some light on that aspect of concern right now.
So your concern is related to stroke diagnoses, vaccine implementation, and thrombotic events after vaccine implementation. Is that right?
Thrombotic events after the vaccines. And right now it's looking like it was just AstraZeneca. And then it became a J&J question. But I'm also raising the issue of perhaps it's all the vaccines and we're not adjudicating with the others. So can you talk a little bit about what we might be seeing in your database? Did you look at it in terms of the vaccines at all? Or any comment on what, and I'm a little off track here, but this is the news of the day. So
those are excellent contemporary questions, Ms. Peterson. Our study period did not cover the contemporary period when vaccines were implemented. Our cutoff date was June 30th, 2020. So we did not evaluate this in our project. However, from a public health perspective, we should be very cautious before we imply causation of vaccine and thrombotic events, because stroke is common, COVID-19 vaccine implementation is common and rampant, and we should be very cautious that even though you see an event after a COVID vaccine dose, that we should not automatically assume that it was related to the vaccine. I think we need to look at national, large source data before we can imply such associations.
Thank you, Dr. Nguyen. I'll also add that perhaps we did not yet have, as a scientific community, ability to flesh out the mechanisms of thrombosis in neither the COVID-19 related stroke nor those that occur in apparently in the follow-up to vaccination. So I think we need real science behind that rather than observation, as Dr. Nguyen suggested.
Absolutely, Dr. Rost.
Okay, I would like to call on our next journalist, Dr. Wilner with Medscape. Could you may activate your audio and ask your question.
Can you hear me?
Yes.
Thank you for asking me to give a question and for a very clear presentation. You mentioned that there were 120,000 COVID hospitalizations during your time period and the stroke rate was 1.5%. What would be the control group or the expectation in a non-COVID population so that we can draw some conclusions from your data?
That's an excellent question. I think you'd have to have a control population before the pandemic to look at what is the overall rate of stroke, and I don't have an answer to your question at this point.
Any follow-up question, Dr. Wilner?
Well, 1.5% doesn't seem that high to me. So I wonder whether that's just the background noise. And I think that's a pretty important point, that there must be some sort of, NIH data. Somebody must have data on hospitalizations and stroke rate just kind of, over time. Because I think if the background rate is, 0.5% or if it's 3%, then that makes a big difference. So it's great to have that single data point, but I think people are going to throw that around without being able to sort of know what it really means.
I think this data point is a function of how readily available testing was for SARS-CoV-2 infection. So if you look at earlier reports of the rate of stroke among COVID hospitalizations, the rate was about 1.1 or 1.2% across wide swaths of patients. But as we progressed, because we were looking out to June 2020, testing became more readily available. So that's why we think we have a higher number than what was previously reported. And obviously, we also saw geographic variation across continents. South America, we saw higher rates of stroke among COVID hospitalized patients. And what explains these differences in rates is very interesting. I don't think we have all the answers to that, other than COVID just being more common in some of these countries. You just see higher rates of stroke. But essentially, overall, globally, our stroke rate was estimated at 1.5%.
Okay, we'll go to the next question. Joseph Elia, can you please unmute yourself, and then you can ask your question. Also, please say which publication you are with.
Hi, this is Joe Elia from the NEJM group. Dr. Nguyen, could you please elaborate on what you mean by recovery? Is this a recovery not of stroke, but of the hospitalizations for stroke? What exactly are you talking about here?
Excellent question, Mr. Elia. Thank you for your presence. The recovery means the recovery of stroke hospitalizations as we moved into the later months of the pandemic, meaning we had 11 to 13% drop early on, but then we saw a 10% increase of stroke admissions in the later months. Did I answer your question properly?
I have muted him, and we are moving on to the next journalist. However, we can have him ask a follow-up question via email afterwards. So Ed Sussman with MedPage Today, you may unmute yourself and ask your question. Are you able to unmute?
I think so.
Yep, there you go.
Okay. I had a few questions. Do you think that you're looking at the area, the time frame from the middle of last year? Do you think things have changed? I know you haven't studied those, but do you think the metrics have changed in the past 6 months, especially as vaccines roll out? And why do you think there were these declines in treatment in those early months of the pandemic? Do you think people were worried about going to the hospitals?
Both excellent questions, Mr. Sussman. Thank you for posing this. In terms of the changes over time, Dr. Noguera is also working on a large cohort that extends to a year out. And we do see changes as a function of the COVID waves, and that remains to be elucidated further once the material matures. We are planning a follow-up study as well to look at this as we roll into the vaccine phases of the pandemic. And then to answer your second question, why do we think these declines occurred? There are so many reasons to share. Fear of contracting coronavirus or nosocomial infection during the hospitalization is cited as a very common reason across many publications. A reduction in exposure to other infections as we wear masks can also potentially reduce our risk of having a stroke with less exposure to infectious events. And then potentially as we shut down elective surgeries and patients are not on blood thinners, then we may have a decline because there are less perioperative related strokes. That was another potential reason cited by one of our co-authors in Australia. So the last reason is thinking, is there a true decline? Was there really a real decline imposed by these lockdown measures, the environmental change of wearing PPE, people adopting better habits? This may not be a negative thing. This may be a positive thing if we think about it the other way, or negatively if people are avoiding stroke care because we can't measure what was not, patients who did not present, then that may be negatively affect overall in the population level. Thank you.
Thank you, Dr. Nguyen. In the interest of time, we do have a couple more questions, but I will connect those journalists with you after the press conference so that we can move on to the next presentation. Thank you to all the journalists who did ask questions. Andy, you may proceed.
Thank you, Renee. I would like to now introduce Dr. Rost's second guest, Dr. Sabrina Paganoni. Dr. Paganoni, you may turn on your video and microphone as we get started here. Dr. Paganoni will now speak about her abstract titled Long-Term Survival of Participants in the Centaur Trial of AMX35 for ALS. This abstract is no longer embargoed. Dr. Paganoni, welcome. Please share with us a summary of the findings of your research.
Thank you. I'm delighted to share the results of the Centaur trial with all of you today. The Centaur trial is a milestone in our fight against ALS because the drug that we tested in the trial is the first to show a combination of both functional and survival benefits for people living with ALS. ALS is also known as Lou Gehrig's disease. People with ALS have progressive muscle weakness, and this means that they lose the ability to do the things that we do every day that we take for granted, like walking or talking. And typically they die within a couple of years from diagnosis. So ALS is really a tragic disease. In the Centaur trial, we tested a new drug for ALS called AMX35. And the excitement is that people who took AMX35 in the trial retained physical functions longer, like speaking, swallowing, and walking. From a patient perspective, this means better function with activities of daily living. When we follow the participants long-term, we found that the risk of death was 44% lower among participants who took AMX35 compared to placebo. And in addition to surviving longer, participants who took AMX35 experienced lower rates of dependence on mechanical ventilation and lower rates of first hospitalization. AMX35 protects against nerve degeneration by using a combination of two medications that each target a different cell component, the mitochondria and the endoplasmic reticulum, respectively, to keep the nerve functioning properly. In the trial, the drug was safe and tolerable. And let me add that AMX35 is very easy to administer, either by mouth or via feeding tube. And in addition, it requires no lab or ECG monitoring. So in summary, the results of the CENTAUR trial provide substantial evidence supporting the role of AMX35 in ALS. If approved, I expect that AMX35 will be widely used for the treatment of ALS. The next steps will depend on discussions with regulatory agencies. Before I end, I would like to thank all the collaborators that made the trial possible, especially Dr. Mary Sukovich, the senior author of the paper, the NILS Consortium, the ALS Association, ALS Finding a Cure, and Amylyx Therapeutics. And finally, I would like to thank all trial participants because without their partnership, ALS research would not move forward. I look forward to presenting all the details at the AAN meeting next week, and I will be happy to take your questions. Thank you.
Thank you so much, Dr. Paganoni, and thank you for the work that you do on this pernicious neurological disease, which really has been a focus of so many efforts to rein it, so to speak, rein it in and have some control over this relentless neurodegeneration. I wanted to ask you with regard to the novel trial design in the way that you've assessed the participants' vital status. I think that really made a difference. And, we often know that methodological advances in the way we look at the data and evaluate participants' outcomes really do matter in the way we're able to uncover layers of complexity in how they respond to the treatments we expose them to. So speak about that. Speak about that novel assessment of the vital status and how is it different from what's been done in the past, and how do you think it may be applied to what we're going to do in assessing outcomes in ALS patients in the future?
Thank you. This is such an important point, and something that's very dear to us. So when we designed the trial, we really wanted to use a patient-centered design. So we kept the randomized phase short, and then we allowed everyone to have access to open label extension, so to active drug long-term. And then we were able to follow the vital status of everyone, so to determine basically their vital status long-term by using publicly available databases. This is actually a process that's commonly used in cancer or other disease areas, but was new to ALS. And because of that, we were able to estimate the long-term survival rates for everyone who entered the trial, even the people that were lost to follow-up. So I think this is really something that, is something that is used in other areas of medicine, and it makes total sense for ALS. And I really think that this will be adopted more widely in ALS studies moving forward.
Thank you very much. I also wanted to ask you with regard to the two components of the AMX35. Obviously, there's a lot of basic science and the science, preclinical science that goes into determining the combinations, et cetera. Do you have any inclinations from what the work that's been done to know that these are very specific combination and whether they're synergistic? I know that they affect two components of the cellular function, both mitochondrial and the neurodegenerative. But do you believe that this is each acting in its own pathway and then they're synergistic, or do you think this is just kind of are they working in parallel and the combinations just been serendipitously discovered?
Now, that's an important point, and we're still doing additional work to better clarify the basic science or mechanisms that support these effects. Now, each individual component had actually been studied in the past, both in the lab and in early phase clinical trials. And even individually, they were shown to have neuroprotective properties. But then when the company combined them in the lab, they showed that their effects were greater when used in combination than when each component was used individually. So this really builds on previous research and sort of adds an additional layer to this, showing that there is even greater impact when the two components are combined. Now, why is that? Well, we don't fully know that, but it does make a lot of sense if you think about the big picture of ALS pathophysiology. So in ALS, there's many different cellular pathways that are affected, and there's many many different causes of ALS, but ultimately they converge on a few key mechanisms, such as mitochondrial dysfunction and endoplasmic reticulum stress. So by blocking distal mechanisms that seem to be common for many people with ALS and really underlie the disease or some of the final steps that are common for many patients and many different forms of the disease, this was probably the combination that we were waiting for to really try to have a better impact.
Wonderful. Thank you so much. The abstract now is open for questions from the floor.
Thank you both, Dr. Rost and Dr. Paganoni. We'll now take questions from journalists. So please put your questions in the Q&A portion, and I will hand it over to Renee if there are any questions at this time.
Okay, thank you very much, Andy. I'm not seeing any questions. Oh, I do have one question that just popped up. And also, if there is anybody out there that has a question, again, please put your name and your publication in the Q&A, and we will then call on you. So, Lynn Peterson with Trends in Medicine, you may ask your question.
Some of these data were announced last fall. And so what is new in this data that hasn't already been released?
Yeah, thank you for your question. Yes, some of the data was presented last fall, both in terms of better function over the 6-month randomized period and our pre-specified survival analysis showing longer survival in the trial. Now, what we're going to present next week are new data related to lower rates of hospitalization and dependence on mechanical ventilation, which are obviously important life events for people with ALS. So we were really pleased to see lower rates of those events in people that receive the active drug. And the other piece of information that's really exciting is an exploratory analysis that we did in collaboration with European researchers looking at observed survival, comparing it to predictive survival in this population. So essentially, we leveraged a model, a prediction model that can generate individual predictions for each participant and estimate their predictive survival based on their baseline characteristics. And when we did that analysis and we compared observed survival to predicted, we saw that the survival benefit was even greater than what we had seen in our original pre-specified analysis, which was great to see.
Okay, let's move on to the next question. Dr. Andrew Wilner of Medscape, if you would like to ask yours, you should be able to unmute yourself now.
Yes, thank you. I guess my concern is that these results are too good to be true. You know, there is this phenomenon that when patients enter clinical trials and they get such great care that they tend to do better than they do just under ordinary treatment. And you mentioned that the randomization period was unusually brief. So I'm wondering, you know, how reliable is the control? Where were the controls? If all the patients were getting the drugs, where's the control group?
Yeah, no, thank you for asking. And that gives me an opportunity to clarify. This was a randomized placebo-controlled trial that lasted 6 months. And that's a period of time that's established to be sufficient for ALS to see changes in function between active and placebo. And so that it's over that period of time that we saw a longer retention of function in people that were randomized to AMX 35 compared to placebo. What we did after that was to allow people who had completed the placebo control period, which was 6 months, to have access to active drug long-term. And then we compared in the pre-specified analysis the people who were originally randomized to AMX 35 to the people who were originally randomized to placebo. However, essentially, because we use this patient-centered design, we essentially think we underestimated or diluted the effect. So I just want to clarify, this was a placebo-controlled trial.
Okay, thank you, Dr. Paganoni. We do have one more question. Joseph Elia, if you would like to ask your question.
Hi, Joe Elia from the NEJM group again. I wanted to ask you, Dr. Paganoni, how many patients were involved with the trial?
137. So that's the number of people that were randomized in the 6-month placebo-controlled trial. And then, as mentioned earlier, we followed the survival, long-term survival for all of them as part of this novel method of using public records to determine survival long-term.
And were they randomized 1-to-1?
No, they were randomized 2-to-1, active
Okay.
and placebo. Yes, it was
Thank you.
published in the New England. Yeah, thank you.
All right, those are the questions that we have. So Andy, if you want to proceed with the first press conference. Thank you.
Excellent. Thank you, Renee. I would now like to introduce our third guest, Dr. Michelle Milke. Dr. Milke will speak about her abstract titled Comparison of Neurofilament Light and Total Tau as Blood-Based Biomarkers of Neurodegeneration Association with Cognition and Neuroimaging Outcomes. Please note that this abstract is emerging science and is embargoed until 12:01 a.m. Eastern Time, Friday, April 16th. Please do not publish stories on this research until the embargo lifts. Hello, Dr. Milke. Please share with us a summary of the findings of your research.
Thank you, Andy. And I'd like to give a big thank you to Dr. Rost and the AAN for the opportunity to present this work today. And I do also want to highlight that the presenter of this abstract is Jordan Marks, who's an MD PhD student. And so he will be giving a brief overview and more depth of the project later on this week. Neurodegeneration, or brain cell loss, is a common characteristic of many neurological disorders, including Alzheimer's disease, vascular dementia, and Lewy body dementia, among others. However, the causes and location of the neurodegeneration can vary. So, for example, in Alzheimer's disease, amyloid plaques and neurofibrillary tangles contribute to neurodegeneration. And there tends to initially be more brain cell loss in the temporal lobes. And vascular-related cognitive impairment, infarcts, white matter hyperintensities, and microbleeds can contribute to cognitive changes. Neurofilament light, or NFL for short, and total tau are two proteins that may be biomarkers of neurodegeneration, and importantly, can be transported and measured in the blood. However, there's limited study of the comparison of these markers for several cognitive and neuroimaging measures. Understanding which marker is associated with which aspect of neurodegeneration will inform how each marker can best be utilized for specific clinical purposes by physicians, and also what information each marker can provide. Therefore, in the current study, we compared the association of plasma total tau and NFL as biomarkers of cognitive decline and with neuroimaging measures of brain atrophy and white matter changes in the community-based Mayo Clinic study of aging. We also replicated our analyses in the Alzheimer's disease neuroimaging initiative to determine whether the value and information provided by these markers varied based on the setting and participant recruitment methodology. We found that at a single point in time, plasma NFL was more strongly associated with all current measures of cognition and brain imaging variables. However, having information on plasma total tau levels did provide some additional information. For example, the combination of having both elevated NFL and total tau was more strongly associated with worse memory performance at the time of assessment. Thus, total tau may add some diagnostic benefit beyond NFL, particularly for neurodegenerative diseases such as AD, which have a primary memory component. We also determined whether these markers could predict change in cognition and neuroimaging variables over a median of about 6 years. We found that NFL was more strongly associated with change in all our outcomes, both cognitive and neuroimaging. And the information provided by total tau in this case did not add any prognostic value. So these results suggest that for prognostic purposes, NFL better predicts the rate of neurodegeneration and cognitive decline, regardless of what the cause of the neurodegeneration might be. And as I mentioned, we did try and replicate these results in the ADNI cohort, and our results were replicated, suggesting that NFL is a better prognostic marker across clinical settings. But at this point, I'd be happy to take questions. Thank you.
Thank you so much, Dr. Milke. First of all, thank you so much for submitting your original work to the AAN annual meeting, particularly in emerging science format, because this is truly what the spirit of emerging science is, kind of hot off the press. So I wanted to ask you, having done myself a lot of work in biomarkers are tricky, but NFL strikes it again out of the park. So I wanted to kind of gain your insight into whether you think it is time we truly think that the NFL, the biology behind NFL as a biomarker is truly representative of neurodegeneration, or do you think it's better as a biomarker, i.e. it has the properties methodologically that we're more able to capture, the robustness of how we're measuring it, or, something else that is driving more of a kind of a methodologic benefits of the biomarker versus the biological one?
So I think that's a good question. I do think that, NFL is biological representative of changes in the brain. So if we take a step back, originally NFL and these markers were looked at in cerebrospinal fluid. And studies have shown that there is actually very good correlation between plasma or serum levels, blood levels of NFL with CSF measures in the brain. The one complication perhaps with NFL is that it is a non-specific biomarker of neurodegeneration. So as I mentioned, we do see it in Alzheimer's disease, in vascular dementia. It's been looked at in stroke, in multiple sclerosis and other diseases as well. So I think, it is more probably indicative of the rate of neurodegeneration, which itself is informative.
Mm-hmm. Thank you. I also noticed that since you focused on NFL and tau, I've noticed that amyloid, beta amyloid, has not been included in this study. Is there a reason why not, or is it just a selection of these markers, or just clarify?
No, thanks for that question, and let me just give a little bit of background again too. So within the Alzheimer's disease field, there is currently the National Institute on Aging Alzheimer's Association criteria, which we refer to oftentimes as the AAN network, which is provides more of a biological definition to Alzheimer's disease. And so within that framework, A stands for amyloid, and biomarkers of amyloid might be amyloid PET, CSF amyloid. Biomarkers of T for tau is phosphorylated tau or tau PET. And then biomarkers of N or neurodegeneration, which are supposed to be more nonspecific, have been proposed as possibly total tau or NFL. So our goal here was really, as I mentioned, neurodegeneration can be caused by a variety of conditions. How do these different so-called neurodegeneration markers match up to cognition? How do they match up to different neuroimaging measures? So we were really trying to understand more of the specific neurodegeneration markers from that standpoint.
All right, great. And one final question for me is that obviously a lot of your work is in MRI-based biomarkers as well. And I wanted to ask you if you feel that there is advanced neuroimaging, using MR technology that is either currently being developed or maybe coming in a few next years that would be even more of a sensitive measure of association between these biomarkers, fluid biomarkers of neurodegeneration versus imaging biomarkers, because again, it's all depends on methodology. And will we go beyond volumetrics? Will we go beyond quantifying, let's say, white matter structural integrity using diffusion images? Is there something in a pipeline you're excited about, which may be a better MR biomarker as well in association with NFL?
So I think, the technology is rapidly advancing, not only for the development of blood-based biomarkers, but also for imaging. And there are a lot of exciting advances. I can't say, for one advance over another. I do think bringing it back to the blood-based biomarkers is that, the MRI is absolutely essential from a neurology perspective and determining ultimate diagnoses. I think the blood-based biomarkers will be more important at the community or potentially primary care level down the road. before we can get to the imaging. And so it may not be perfect, but it's a possibility of screening those and identifying those at risk.
Wonderful. Thank you so much. This abstract is now open for questions.
Thank you both. Excuse me. Again, please note that Dr. Milke's abstract is embargoed until 12:01 a.m. Eastern Time, Friday, April 16th, 2021. We'll now take questions from journalists through the Q&A function. Renee?
Thank you, Andy. Yes, well, I'm watching to see if anybody posts their name and their publication. And if you do, I will call on you to answer or to ask your question. At this point, we don't have anybody posting questions on this. We'll wait one more second. I think we can proceed, Andy. No questions from the journalists.
Okay, thank you. Dr. Rost, would you, do you have any closing remarks you'd like to share?
Sure. Well, I hope that you enjoyed our virtual Top Science Conference. This is an unusual format, which I hope we won't have to repeat in the coming years, but if we do, we'll be ready, and we now have our trial run. And I'd like to thank the presenters for doing an exciting science at the times when it's most challenging, and to all of the journalists who maintain their continuous interest in science, again, under the duress. And I hope that you all have an opportunity to learn about the entire breadth of the AAN scientific program throughout our upcoming meeting. And we'll see you in the virtual hallways of the annual meeting later this week. Thank you, everybody.
Yes, thank you, Dr. Rost, and to the journalists. If you still have any questions for the authors, please email our AAN Annual Meeting press room team, Renee Tessman and M.A. Roscoe, at the email addresses shown on the screen. On behalf of the American Academy of Neurology, I'd like to thank Dr. Rost, as well as Dr. Nguyen, Dr. Paganoni, and Dr. Milke for sharing their research today. Thank you to all the journalists in attendance for joining us for our celebration of science. For more information about the American Academy of Neurology and the 2021 AAN Annual Meeting, visit AAN.com. Thank you.
Thank you, everybody.